Okay, let's start with our next session. It's a pleasure to invite Nir Ben-Tal, and he will tell us how we can improve the function annotation of proteins.
So the title of the talk is From the Detection of Functional Regions Towards Function Annotation in Proteins, and it's not, we are not still there, it's just towards, okay? So the work was done mostly by a guy named Rod, who is here in the audience. Maybe you can stand up. And that means that if we have, and others have helped, but that means, since he's here, if there are really technical questions, we can even hope to get answers for these. So the background is that, as we all know, we have the Structured Genomics initiative going on around the world, which means that, since one of the goals is to map the protein fold space, it means that we have a lot of proteins with known structure but unknown function. We call them hypothetical proteins, and not necessarily the best name, but that's what they call them. And here are examples of hypothetical proteins, so they come in different types and different shapes, and the point is that we don't know what's their function. And so, and the idea is to try to develop computational tools to predict their function. And the typical methods are based on sequence homology, for example, BLAST, or as we heard, context-specific BLAST, which probably would do a better job in that, or the detection of specific sequence motifs that are indicative of a specific function, as in ProCyte and similar facilities. There is also the possibility of the detection of structural similarity, and some people are using global structural similarity, as in DALI, etc., to deduce the function. And others use local structural similarity. We are leaning towards this end of the scale, and of course there are methods that combine both sequence and structural similarity. So our assumption is that structure is essentially, that function is a local property. If a protein should bind DNA, then there are specific amino acids that recognize DNA, and presumably if we detect these, then we know that, and they are in structural proximity to each other, and then if we detect these, we can deduce the function. For that, we start by the detection of any functional important amino acid, and we use the conserve server for that, which we developed several years ago. This is an example of that. You see a protein that, in this case, it's a known case because we already see the functional region. That's where the yellow molecule is. But if we map evolutionary conservation as if we did not know where the functional region is, using the conserve server, where variable amino acids are in blue and conserved are in purple, we see that the eye is drawn to the peptide binding site in this case because it's highly conserved. So even if we did not see the peptide, we knew that this is the functional region. And the question now is, since we usually can detect this region, the question is whether we can derive information from this region to understand what the function might be. And of course, we are not the only one to do that. There are many other groups, including a lot of you in this audience. So we detect the functional region and try to match it with a function, and this is actually done using by going over all possible patches of amino acids that are conserved, and based on some maximum likelihood principle, we give confidence that this region is actually the interesting region of the protein. And with this tool, which was developed, we derived a data set of over 700 proteins of known structure, but no functional annotation. and it's reported in this publication. Now, 90% of the structures were actually solved in structural genomics initiatives. Others were solved in other facilities. And the functional region of each of the protein was predicted by this tool, the PatchFinder, and that's a starting point, as I said, hopefully to annotate the function of the proteins. And the data set is available here, and I urge you to look at this data set, because I believe that with some of the tools that are developed in this room, it's possible to annotate some of these proteins already, perhaps by taking into account the patches of evolutionary conserved amino acids. This is one example. We do not have an automatic version to annotate function yet of all the proteins. However, we did look at several cases to try to see what we can do by combining other tools with manual use of other tools. So this is an example. This is the functional region, presumed functional region of this protein. This is the maximum likelihood of the protein. We actually give confidence to the prediction based on the degree of conservation and the size of the patch. And in this case, you can see that there is high confidence in the fact that this is a functional region. There's another patch with low confidence, but I don't present it here. Another indication that it can be a functional region really is its location. It's in a concave region, where one could imagine that it would a ligand could fit here nicely, and indeed that's going to be the case. So Guy looked at several computational tools, including the set of tools that is found in ProFunc, and it found local structural similarity between this protein and the ligand binding site of a GTP binding protein from the human protein RAB5A. And when you superimpose the local similarity, you actually see that there is also a certain level of global similarity between the structures. And that protein was crystallized in complex with the substrate, GTP analog in this case. And you see, if you would imagine docking this analog into this pocket, you see that it would fit nicely. And indeed, Guy tested, and there is very little clashes if you do that. So the suggestion is that this protein may bind a GTP or GTP analog or something like that. And in principle, one can imagine going over many other predictions and predicting what they might bind to. However, Guy decided to take a slightly different route and started to look for DNA binding proteins based on in the N-Func database. And for that, he developed a methodology to detect DNA binding proteins based on their evolutionary conservation of the maximum likelihood patch and other properties. So again, we are not the first to have done this, and others have done it too. So it's important to do it. We know that an estimate of something like 12% of the proteins in the human genomes are DNA binders. They are involved in a variety of physiological processes in DNA transcription, maintenance, replication, etc. This is an example. It's a Tata box binding protein, and you see the DNA here in yellow and the protein itself in green. And here are several examples of DNA binding protein, including the one that I showed. You see that they come with different shapes and various modes of binding to the DNA, using various motifs to detect the DNA. And the goal in this project is dual. So the first is to annotate DNA binding protein, that is to go to this data set of 700 proteins or so, and to look for DNA binders, and also to identify the DNA binding sites. Now, of course, one possibility is to try to train some machinery to detect known motifs that bind DNA, and people have done it, and I'll go over it soon. But Guy aimed at something slightly different. The thought was that maybe there are still modes of binding to DNA which are missing, which we don't know anything about. So he tried to generalize things about the binding site and other properties of the protein, and based on that, possibly be able to detect new modes of binding to the DNA. So previous efforts relied on, for example, on the electrostatic characters of the system. So the DNA is negatively charged, and therefore the proteins that bind DNA are often positively charged, and people have used that. As I said, there are other methods that are based on the detection of specific structural motifs, such as the helix-turn-helix motif here. And these also work well for the detection of DNA binders using these motifs. The geometric attributes of the protein surface was also used. Amino acid composition and amino acid nucleotide interaction preference, like pair potentials, were also used. And Guy decided to use, as I said, the local region of binding to the DNA, as well as other properties, as I said. So, but before that, he analyzed the local structures, these functionally important regions, the maximum likelihood patches, if you want. One of them is marked here in red, in red, sorry. And he's to see what's the degree of overlap between these and the DNA binding region. So typically, so first of all, the vast majority of the maximum likelihood patches are composed of DNA binding residues, and typically they cover half or less than half of the DNA binding residues. So we don't cover the entire protein DNA interface, and I'll get back to this soon. The maximum likelihood patches are enriched with hydrogen bonds with the DNA, which are important for the recognition, and that suggests that the patches that the guy finds typically are the region that determines specificity in the binding to the DNA. The question is how we deal with conformational changes, because very often protein may change conformation upon binding to the DNA. And the question is how is the maximum likelihood patch depends on the conformational changes, and in extreme cases, of course, there are, and that's a continuation of the previous talks, that there are unstructured regions that gain their structure upon binding DNA. So I'm not talking about such extreme cases. I'm talking about cases where we know the structure of the bound and unbound protein, and Guy collected 13 such cases and showed that for the most part the maximum likelihood patch looks very similar. This is an example. This is the worst case where the structural change is really big. You see the unbound form in green and the bound form in blue. And you see that there is a big loop that changes conformation to facilitate the interaction with this adenosine basis. And you see how large the change is. And nevertheless, the maximum likelihood patch is essentially identical to the one bound and unbound. So which features Guy added to the classification? So first of all was the maximum likelihood patch, and then also the electrostatic potential, the secondary structure content, because certain specific motifs, for example, are based on alpha helical structure, hydrogen donors and acceptors for the specific interactions, the dipole moment, and spatial asymmetry in the distribution of arginine, glycine, asparagine, and serine, and the percentage of these residues in the protein and in the binding site. Also the amino acid conservation pattern, which was a major factor in the analysis. And here you see that in some cases he included it, the property on the maximum likelihood patch, and in others in the entire structure, and sometimes the property was included in both. and in general, so I'm not going in detail over all this, but because it appears in the publication. But the classifier achieved a sensitivity of 90% and specificity of 72% on a data set that we consider realistic, in that it contains about 15% DNA binding proteins, realistic for when you analyze a whole genome or something. It outperformed related machine learning based methods, and you see here the main attributes for the success. So the calculation here was done by running the method with and without the property. So for example, the major attribute is the electrostatic potential you see here, amino acid conservation patterns, secondary structure content, amino acid content are all important, and it's listed here. And another source of confusion may be the distinction between DNA and RNA binding proteins. And the question is, can we discriminate between them? So RNA has, the reason is that RNA has a physicochemical properties that are very similar to DNA, and the interactions of the protein with the RNA and DNA are similar too. And in fact, there are examples of a protein that bind both. One of them is shown here. And indeed, we have a problem, and we confuse DNA and RNA bindings with the current protocol. However, Guy just told me that preliminary analysis that he did where he addressed only the question of, let's say that we know that a protein may bind either DNA or RNA, can we classify, and there we do better than random. But it's work in progress. So the current thing confuses DNA and RNA binders. And of course, the initial motivation was to actually detect DNA binding proteins in the N-Func database. And when Guy applied the classifier in N-Func, over 200 proteins were predicted to bind DNA, which is a lot, but that's expected. So based on the false discovery rate, this is a kind of that's the kind of number that you would expect. So presumably, a lot of the prediction are false positive predictions, up to 2/3 of them. But there's also the 1/3 which is presumably real DNA binders. And that's much better than what you would get by chance alone. Like by chance you would get only 15% or less of protein. So now when experimentalist wants to look for DNA binders, then applying the predictor increases the chance of predicting of hitting a real DNA binder significantly. He also analyzed specific cases just to make sure that there is something in it, and this is one example, entry 2FNA. You see here it binds a nucleotide, and we believe that here it binds DNA. You will see it in a minute. So when he looked in other resources, he found global similarity. That was through ProFunc. He found global similarity to another protein, CDC6, and you see how well they superimpose. And CDC6 was a complex that binds DNA, so presumably also 2FNA binds DNA. So in this case, we should have predicted that the protein probably shouldn't have been in the database in the first place, but it's difficult to detect it without the superimposition, which was not trivial to do first. So that's an example where the prediction was probably correct. So I'd like to conclude and say that PatchFinder finds the core of the DNA binding site, and the evidence there are from the fact that there are a lot of hydrogen bonds and that it's not the entire site. In fact, if you think of that, so current models for protein binding to DNA are based on the assumption that the protein interact generally with DNA, maybe with electrostatic interaction predominantly, and then it slides along the DNA and hops between the various strands of the DNA until it reaches its location of function. So we believe that the maximum likelihood patch is responsible for the latter stage in the binding, where the DNA where the protein recognizes the exact location on the DNA. So these regions, as I said, are enriched with hydrogen bonds, which are typically important for recognition. PatchFinder is not meant to detect the entire DNA binding region. There are better tools for that, and here are references. But it's good in discriminating between DNA binders and non-binders. So we reached a sensitivity of 90% and specificity of 72% on database that represent the expected user data. And the anticipation is because none of the known motifs were taken explicitly into account in the learning process, we believe that with this tool it's possible to detect other modes of binding to DNA, presumably using other binding motifs. So the future directions, we would like to examine experimentally the prediction of the DNA binders, and hopefully Guy will do it in Yanai of France lab, who is now also an experimental lab. And also we would like, as I said, to distinguish better between DNA and RNA binders. And this is just a general approach that shows that you can start from local descriptor of the binders to detect the function, and we hope that it's possible to derive other PatchFinder-like tools to detect functional classes of protein. And I'd like to thank the people who did the work. As I said, it's mostly Guy who's here. Thank you very much. I'll be happy to answer questions.
There's time for one or two questions. Go to the microphone, yeah.
As far as the DNA binding is concerned, I believe that perhaps the flexibility of the site might be taken into account without explicitly modeling that. But in principle, this could be very different for a number of other proteins, especially multi-domain proteins. So perhaps you could actually enrich your database by considering where there are hinge regions or multi-domain proteins, by considering normal mode analysis or some sort of a better exploration of the possible binding site, right?
Yeah, that's a good suggestion. Thank you.